We are going to start on dihybrid crosses today. So they're a little more complicated than the monohybrid crosses, but I think you can do them if you just get you a piece of paper and a pencil and follow along. And if you get stuck, feel free to pause, stop the video, go forward, go backward, or email me, text me, and we'll see what we can do to help you along your way. Okay, so dihybrid crosses. Well, hello, ESPN. Okay, so di means two and hybrid means a cross. So we're going to be looking at two traits today, like a tall plant with purple flowers and a short plant with white flowers or something like that. So two different traits on the same parent. Okay, so the first thing we want to look at is make sure you understand that we're talking about two traits, which means we have two alleles for each trait. And if we're looking at two traits, we should end up with 4 alleles. So if we're looking at yellow and green peas, yellow is dominant to green, and then we're going to also look at texture. So we would have round or wrinkled peas. So yellow and green and round and wrinkled are the two traits that we're going to look at today for notes. All right, so here we go.
I hope you two will be really happy together.
See, I hope you two will be really happy together. I didn't know what it said. All right, so here we go. Big R indicates, so if we're looking at it, let's mark this up a little bit. So we're looking at two traits, big R here, and then big Y here. And then on this plant, also, little r, and little y. Okay, so let's see. Here's our next thing. So we have a homozygous by homozygous dihybrid cross. We're going to cross round here, yellow peas, and we're going to cross them with wrinkled green peas. So the two dominant traits crossed with the two recessive traits. To do that, we have to determine our gametes. Now before, when you had big R, you only had two options, right? You had big R or big R. Well now, because we have the second trait on here, we're going to have 4 options, and we're going to have to do something called FOIL. Okay, so to do FOIL, let's start with, here, let me put it over here. FOIL, F-O-I and L. Okay, and so we're going to have FOIL first. Here we're going to have first. Okay, so when we do first, we're looking at the first R and the first Y. So my arrows are off a little bit here, but you can see big R, big Y, and the first gamete would be big R, big Y. Okay, then the second gamete, we're going to do the two outside letters. Okay, so let's put outside here so you know what that stands for. All right. So the green would go with the two outside letters. So this R here and this Y here. And so our next gamete would be big R, big Y. Then we're going to deal with the two inside letters. So the I stands for inside. All right. And we're going to look at this R here and this Y here. When you put those two together, of course, you know, because the gametes are all the same, we're going to end up with big R, big Y. And then the last two letters, that's what that L stands for, is the last. So the last R here and the last Y here. So this one with this one. And we're going to end up again with our big R, big Y. Sorry about my arrows. That's an old PowerPoint. So first, outside, inside, last. FOIL. I think it's an algebra thing that you guys do, so you should already know how to do it. All right, so let's clean everything off of this page and go to the next page. All right, so because we have 4 gametes for each parent, we have to have 4 rows and 4 columns, so that's a 16 square. Okay, so let's look at what this would look like. All right, so we have our big R, big Y, big R, big Y. This one should be over here. And then down this way. All right, so the simplest way to do this is just like we did before. We're going to bring down this letter into this square and this letter into this square. And then we're going to bring this letter over and this letter over. Now remember, capital letter first, like letters together in the order of the parents. So to do this, in this square, you're going to have big R, big Y, little y. Very simple. So if you were going to bring this one all the way over and this one down, in this square, you would have the same thing. All right. And because the parents are similar, so all of these gametes are the same and all of these are the same, we could easily predict that everybody in the squares are going to be heterozygous. So go ahead and fill in your squares. Okay, so now let's talk about this. Big R indicates that this one is round. So if we were going to put round, we would do, let's see, I can write it, I think. So let's do round. Okay, we know it's round because of the big R. We know that it is yellow because it has a capital Y. So big R, big Y is round and yellow. So every one of these offspring are going to be round and yellow, which is going to make us have what we would say as 100% heterozygous and 100% round and yellow. So this heterozygous is our genotype and round and yellow is our phenotype. Okay, so now let's try something a little harder. We're going to take the offspring, so the F1 generation of the cross that we just did, and we're going to cross them again. So now we're going to look at heterozygous, both parents, at both traits. So we're going to do a heterozygous by heterozygous dihybrid cross. So again, the first thing we have to do is we have to determine our gametes. Okay, so to do that, we're just going to FOIL again, very simple. And here we go. And now remember my arrows are off a little bit. So I'll draw it real quick before we do it. So we're going to do big R, big Y. Then we're going to do big R, little y. Then we're going to do little r, little y. And little r, little y. So when you're done, you should end up, let's switch it back now, with something that looks like this. Big R, big Y. Big R, little y. Little r, big Y, and little r, little y. Okay, so now that we have our gametes, we're going to go ahead and we're going to make our 16 square. Now remember, our parents are the same, so you're going to have these gametes on one side of your 16 square and these on the top of your 16 square. Should look something like this. Okay, just like before, we're going to fill in our squares, all right? So if we put big R here and big Y, it should look like this, right? Okay, so here's what I want you to do. I want you to pause the video. I want you to go through and I want you to try and fill in your squares and then come back to the video and see if your squares match my squares. Okay, while you're doing that, I am going to be filling in my squares so that we can take a look at what we've got in here. All right, what you're going to discover is if you look, we have 16 squares, so we have to account for phenotypes and genotypes in 16 squares. Now we know we only have, what, 4 genotypes, right? So we should have dominant, right? Okay, so we could have dominant would be round and yellow. All right, so let's look at these. So you'll see I've color-coded on my PowerPoint, so I kind of gave it away. But let's go through and let's number all of the ones that have a big R and a big Y. It doesn't have to have 2, just has to have the 1. So that would have the dominant trait. Okay, let's see. Here we go. All right, so this one is going to be round and yellow. This one also round and yellow. This one also round and yellow. What about this one? Yep. Round and yellow, very easy. Okay, this one, again, we have a big R, big Y, so this one's round and yellow. This one has a big R, but it does not have a big Y, so this one is not round and yellow. This one is going to be round and green, so it's going to have a little bit of a different thing, so we'll come back and count it in a minute. Okay, what about this one? Yep, round and yellow, so we're looking at 6. This one, no, because that's supposed to be a little Y. What about this one? Yep. So that's 7. What about this one? Yep. 8. What about this one? No, it is not. You're right. It's 2 little r's, so it is not round. It is wrinkled. Okay, and so again, wrinkled here. What about this one? That is correct. That one is round and yellow. This one? Nope, nope, nope. Okay, so out of 16 squares, 9 of them, if we were to mark them where I've numbered, it's a pretty nice little pattern, isn't it? That's right. So 9/16 are going to be dominant and dominant. Okay, so both traits are dominant. All right, so now let's look. Let's go back to this one right here. This one is round, so we know that it's round, but it is also 2 little y's, which makes it green. So this one is our first of our green and round. What about this one? So 1 big R makes it round, and then 2 little y's makes it green. So here we go. Round and green. And then we've got one more right here that is also round and green. So we have 3 of them. Here, we'll make a line this way on these that are round and green. So that's going to give us a 3/16 ratio of round. So dominant for the first trait, but recessive for the second trait. All right. Okay, what about this one? What do you think this one is? Yeah, so these 2 little R's would make it wrinkled, right? 2 big Y's make it yellow. So we now have wrinkled and yellow. So we'll do those with the ABCs. Wrinkled and yellow. So, and this one too, right? All right, so we have 3 because this one's different. So we have 3 again. So 3 of 16 that this time are going to have the recessive trait first and the dominant trait second. So we got dominant, recessive, recessive, dominant, and then our last one, it's all by itself down here, is going to be wrinkled and it's going to be green. So it's 1/16 and it's recessive and recessive. Why did I go through all that? Because the big deal is you're going to have to be able to predict the phenotypic ratio of a heterozygous by heterozygous dihybrid cross. And it's really easy. It's always the same. Here, let me erase these. Okay, it's always the same. And it is going to be what we call a 9:3 to 3:1 ratio. So the phenotypic ratio of a dihybrid cross is 9:3 to 3:1. So 9/16 of them are round and yellow. 3 are round and green, wrinkled and yellow are 3, and wrinkled and green are 1. So 9:3 to 3:1 ratio. Or you can think of these as fractions too. So if they were fractions, they would be 9/16, 3/16, and again 3/16. And then finally at the bottom, we've got that 1/16. And if you add these numbers up, they're going to come up to be 16 of 16. So we've accounted for all our squares. Okay, now let's talk about our genotypic ratio. That one's a little more difficult, and most things are not going to ask you, but it's very simple. All you would do is go back through your squares, and you would look at the different genotypes and write them down. So you're going to have, let me see, 9 different genotypes, I believe. Let's see. Yep, 1, 2, 3, 4, 5, 6, 7, 8, 9. So you're going to end up with a 1:2 to 1 to 2 to 4 to 2 to 1 to 2 to 1 ratio, which is fun to say, but you're not going to do genotypic ratios very often. You might have to count them up, but not very often. So that is how you do a dihybrid cross. That's a lot, I know. So I put 2 worksheets on here for you to practice with. They're both right here. The dihybrid practice worksheet is very long, but it's a lot of practice. So do the emoji one first. It's easier and you get to look at the little emoji ones, but I really do want you to do the dihybrid one. We will not be doing this lab. It's counting corn and it's just really hard to do virtually. So we just won't do it. We're going to do these 2 worksheets, and once you've got it, then we'll move on to ABO blood typing, which is kind of a cool little thing. And then we'll do some pedigrees, and you'll get to ask everybody in your family some questions about, I don't know, earlobes or hairy knuckles or something. We'll figure something out. So until the next time, that's it, and I will see you next week.